Alright, so we're going to be talking about human chromosomes, nondisjunction, karyotypes, and genetic disorders. Alright, really quickly. Remember, chromosomes are made of DNA. You're only going to see chromosomes when the DNA is being organized. We talked about the cell cycle, when the DNA is being sorted out into two piles. And then those two piles will become have a nucleus wrapped around them, and then it'll become two different cells. That's when you see chromosomes, when DNA is being organized or sorted out. DNA is chromatin when the DNA is being used, either used to make a phenotype or it's being copied during the S phase. So that's when we see chromatin and chromosomes. Remember, genes are sections of DNA that produce a trait. Genes are found linked together, one after another, in certain patterns in organisms, and that is what a chromosome is. A chromosome is just a long chain of genes linked together. So is chromatin, but the chromosomes, we can see them more distinctly when we look through the microscope. Scientists think we have 20-some thousand different genes. Well, you inherited those as 23 pairs of chromosomes. That's how that DNA was organized when it was passed down to you. Okay, we've seen a picture like this before. All right, so from looking at karyotypes, we know that humans have 46 chromosomes in every body cell. You may have the number 23 in your head. That's fine, because we have 23 pairs, one member of each pair from each parent. Different organisms would have different amounts of chromosomes, and the number of chromosomes does not dictate how complex the creature is. Hermit crabs have way more chromosomes than we do, so do certain plants. It doesn't mean they're more complicated, it's just they have more chromosomes, and we'll talk about that once we study DNA in more detail. Those other two things, the autosomes and sex chromosomes, I'm going to skip ahead a little bit. When you look at a karyotype, the autosomes are the first 44 or first 22 pairs of chromosomes. Let me take a look at this picture here. That last set, those are called the sex chromosomes, and that's the last two chromosomes, or that 23rd pair, that's the sex chromosomes. That's what that statement was referring to back up here. Okay, so pause this video when you need to, but I'm going to keep moving forward here. A karyotype is a photograph of chromosomes that was then moved into Photoshop or some other photo editing software to group the chromosomes into these things called homologous pairs. You all know that whenever you see the prefix homo, it means the same. So we have pairs of chromosomes that have sameness to them. We're going to take a look at that in just a second. A scientist would make a karyotype because they are suspecting that there is an irregularity with the chromosomes themselves or with the number of chromosomes. So here's a picture taken probably during prophase of the cell cycle. We know that it's after S phase because the chromosomes are doubled, so we know they've been copied, and that's why they look like X's. And it's before anaphase because during anaphase, these would be pulled away from one another. So we know that that's what this picture, that's what was going on. And this would have to, like I said, be moved into some photo editing software to make them into the karyotype image. Okay, these are some things that we were saying already. So this is the karyotype, taking that original image and sorting them out. And we can see that in a karyotype, chromosomes are grouped from the longest chromosomes to the shortest, with those sex chromosomes at the end. You can notice that each pair really look the same with their striping pattern and their size. Chromosomes being bent like this one is really doesn't matter, so we're not looking for that. And like we said before, the first 44 chromosomes are called autosomes. Those last two are called sex chromosomes. And each pair is called a homologous pair. Homologous chromosomes have the same genes in the same order, but they may have different alleles. We know that one member of each homologous pair was from mom and the other was from dad. We don't know which one was from mom and which one was from dad. We can't tell genes based on this. We can't tell if this person has curly hair. All we can see is, are there 46 there? Is there any damage to any of them? And that's really it. Okay? We can also tell gender. If you look at this one down here at these sex chromosomes. Sorry, I was trying to figure out where the pen is here. I don't know. But way down at the end there, those two look the same. Those are two X chromosomes, so this individual is female. If there was a large chromosome with a very small one next to it, that'd be a male because it'd be an X chromosome with a Y chromosome. The X and Y do not look like X's and Y's. It's just the name of them. Okay, so we had already said this. All you're responsible for, my class, is knowing, are there missing or extra chromosomes? Is there some number other than 46? And if we haven't already, we'll be practicing looking at karyotypes. So we're going to be talking about a few different ways that people can have disorders based on the DNA they inherited from their mom and dad. The first type of disorder is going to be called a chromosomal disorder. And that means, again, they have a number of chromosomes other than 46. The way that would happen would be from a random error when the egg and sperm were being formed. Remember, during anaphase, we'd be talking anaphase of the cell cycle, during mitosis, anaphase 1 of meiosis 1, anaphase 2 of meiosis 2, that DNA is supposed to separate away from the other DNA. When it happens nice and evenly, we call that a disjunction, a coming apart. When it doesn't happen evenly, we call that non-disjunction. And I think of pulling on a wishbone. Whenever you pull on a wishbone, somebody gets the bigger part, someone gets the smaller part. That's non-disjunction. It was an uneven splitting. Okay, so this is everything that I had just said. Non-disjunction causes the egg or the sperm to have an abnormal amount of DNA. That does not affect the parents because their somatic cells are just fine. That affects the offspring they're going to be made from those cells. And we're going to take a look at that in just a minute here. Okay, we've said this already, so if you need to pause this, that's fine. But I'm going to keep going because I've already said this. Okay, on the left, it says normal meiosis. And if you look up top, we have a homologous pair of chromosomes. During meiosis 1, they should separate from each other and go into two separate 2-end cells, which they have. And then during meiosis 2, those X's should pull apart at the centromere, and each gamete should have what looks like one chromatid, even though we call it a chromosome at this point. If you look at the picture on the right, we see a non-disjunction during meiosis 1, a not coming apart properly at meiosis 1. We see both of those homologous chromosomes went to the cell on the left. They did not separate from one another. The cell on the right did not receive a copy. And then meiosis 2 actually went normally. It just started with the wrong information. Okay, so this is meiosis 1. Down here is meiosis 2. They did pull apart evenly, but there was just too much DNA in this original cell right here. Okay, so that was a problem. Now, if we think about this logically, say those were eggs, because they sort of look like eggs because they're round. Say a sperm cell came in, and it had its normal that's supposed to be a chromosome, one chromosome here, right? If it combines with that cell or that cell, you're going to have 3 copies of that chromosome. 1 from the sperm and 2 from the misformed egg. If it combines with that cell or that cell, you're going to have a monosome. You're going to have the 1 copy from the sperm cell. Now remember, sperm cells can undergo a non-disjunction and not be formed properly as well. Okay? So those 2 words I just used, trisomy, tri means 3. There's 3 copies of a chromosome instead of the normal 2. A monosomy, there's 1 copy of that chromosome instead of the normal 2. So that's what those words mean. Okay, so these are some things that we had talked about. Wait, sorry. Yeah, there we go. Let me stop here for a second. Um, so again, we see normal meiosis in the picture on the right, on the left. On the right, we see a non-disjunction event. All right. So just some notes here you may not have. I know we might not know what mRNA is yet. We'll get there. But that's what comes from the DNA. So having too many chromosomes, too much of this mRNA, which means too much protein, which is going to cause some problems. Okay? Not having enough chromosomes, won't have enough of this stuff called mRNA, which means we'll have a protein deficiency, and that's going to cause problems too. Remember, every cell of the body is going to have this problem because that fusion of that sperm and the egg, that was the first cell of the body. Every body cell came from that original cell as it went through the cell cycle. So every cell of the body has this abnormal amount of DNA. Okay, and this is a karyotype of really one of the more famous of these rare conditions called Down syndrome. It's where there's a trisomy at the 21st position, and this individual is a male. So we see that because this doesn't affect the sex chromosomes, males or females can get Down syndrome because this is an autosomal trisomy at that 21st position. And if we haven't already, we'll be practicing some of these. Okay, and I'm just going to skip through these because if we haven't seen these yet, we will. This is a process called amniocentesis. And amniocentesis would be if a doctor suspected something like Down syndrome, one of these abnormal amount of chromosome disorders, they could do a procedure where they remove some of that amniotic fluid, which contains some of the baby's skin cells floating around. They could do a karyotype to it to determine, are some of these irregularities a doctor seeing, are they due to an irregular amount of chromosomes, or are they due to some other disorder? All right. So please feel free to rewind. Please write down some questions for me and rewatch what you may need to rewatch.